All right, guys, how are you doing today? We are going to be going over protein synthesis, and this cartoon that you see right here is a ribosome pirate, and of course, all he can make is arginine, so arg, arg, arg. Now, let's take a look at this sheet right, or not this sheet, this diagram right here. So there was this really important experiment done by Beadle&Tatum. Okay, that was their name. Oop, won't let me write over there. Tatum, like Channing Tatum. Okay, Beadle&Tatum, what they figured out, and write this down, this is called the central dogma of biology. Okay, now dogma means like a truth, like a truth we hold dear. And so what this is that one gene codes for one, not for one protein. Okay, and so that seems pretty simple to us now. Yeah, genes code for proteins, we know this, but there was a time when this wasn't known, and it really wasn't that long ago. And so what they did was they grew bacteria. So these were, I believe they were E. coli. But anyways, they grew them on this media, and so this is kind of like some of that jello-y agar stuff, okay? And so the most minimal type of food that you can give these bacteria is called minimal medium. So you're not giving them anything other than like just some vitamins, some salts, you know, just very basic starting materials, okay? Okay, now what they did was they took the wild type, so this is just what we find in the wild for these bacteria, and they grew them on minimal media. Wild type bacteria can grow no problem with these basic starting materials as long as everything is functioning well. Now, what they did was they shined UV light on bacteria, okay? So they took some bacterial colonies and they shined UV light. And as we know, UV light is not good for you. It's a type of radiation that can cause mutations. And so they shined the UV light in hopes of mutating these guys and seeing what would happen. And so they basically tried to grow these mutants on minimal media and nothing grew. So they knew they must be a mutant because they're unable to now grow on this stuff, right? Okay. Now, let's look at this first class of mutants. They were, however, able to grow if you gave them this substance called ornithine. So when you fed these guys ornithine, they grew, okay? When you fed them citrulline, they grew. Now, arginine is kind of this is an amino acid, right? Okay, and it's non-essential. Okay, and what that means is we can make it. We can make it ourselves. Okay, so arginine is an amino acid that not only do we as humans not need in our food because we can make it ourselves, but bacteria are the same way. So here's the thing. Here's what they figured. When we add ornithine, these guys grow. Now, these other types of mutants, they don't grow on minimal media or ornithine, but they do grow when you put them on citrulline, okay? And these ones here, the class 3 mutants, they don't grow on anything unless you give them arginine, all right? So what they figured was there was a pathway, okay? So in a healthy individual, you would be able to take minimal media, okay? You would be able to turn that into ornithine, okay? So there's an enzyme 1, okay? Which means if there's an enzyme, they figured there must be a gene 1, okay? Codes for this enzyme, and that enzyme turns minimal media into ornithine. Okay, there was an enzyme 2, coded for by gene 2, that is going to turn ornithine into citrulline. Okay, and then enzyme 3, coded for by gene 3, and let me go down this way, is going to turn into arginine. Okay, and so they figured this was the biochemical pathway of producing this final product arginine. And so let's look at our class 1 mutants. They don't grow on minimal media, okay? But they do grow when you add ornithine. So if you give them ornithine, they are able to produce citrulline. They're able to produce arginine from there. So Beadle and Tatum concluded that this class 1 mutant has a defect in gene number 1. Okay, so this gene is not functioning, all right, which means they can't make this protein, which means they're not going to be able to produce ornithine. But if you just give the bacteria ornithine, it doesn't really matter if these are being used or not. It can go ahead and make arginine from there. Okay, now class 3 mutants, you can see, they don't grow on any of these. They only grow if you give them arginine. So I want you to think about which gene is mutated for class 3. Think about it, which gene is mutated for class 2. All right? So they figured when we shine the UV light, we're interrupting one of these genes. And one of these genes codes for one of these enzymes in the pathway. And so this was a very important discovery, and I want you guys to remember their names, Beadle and Tatum, one gene, one protein. Okay? So let's carry on. Now, we know now exactly how it happens. We now know that the DNA is unzipped, okay? So the DNA at the gene that we want to make is copied, okay? And so there's our mRNA copy. So we always copy one section of the DNA. That mRNA is going to travel out to the ribosome, and we're going to be able to produce proteins, okay? So proteins are released by the ribosome using the mRNA template, okay? And so ribosomes can continue to make proteins, you know, as they copy the gene, okay? So each gene gets copied, goes out to the ribosome, and then puts together the protein.